Hello students, you are welcome once again to the genetics series. We're going to continue in our series of studies as we look at ways of improving animals through the application of genetic principles. So today we're looking at Module 17 and the title is Animal Improvement and the Application of Genetic Principles. So the knowledge of animal breeding and genetics has been greatly applied to bring about improvement in economically important traits in farm animals. And this knowledge is via selection and or crossbreeding of farm animals. So we'll take a look at each of them. Selection, as we are aware, is the choosing of individuals that will be used as parents of the next generation. And this choice is based on the possession of certain desirable phenotypes or character, which are a reflection of the genetic makeup of the animal. So selection is therefore used to increase the frequency of desirable animal traits or genes in animals. Crossbreeding enables the exploitation of heterosis or what is known as hybrid vigor. Recall that heterosis is an estimation of the performance of progenies, which is usually above the mean performance of both of their parents. So the knowledge of animal breeding has also been applied to the following areas. It is used in improving reproductive efficiency, and this is through the use of knowledge of genetics, especially as it applies to biotechnology in the following areas. It is used in monitoring of reproductive functions and the endocrine status of farm animals. And with this knowledge, farmers can know when to breed their animals so that they can achieve the best reproductive efficiency. Estrus detection and leads to greater control of the reproductive processes of farm animals. Also, we can now carry out controlled mating, especially through the use of artificial insemination. We can also do pregnancy diagnosis very easily. At the same time, we can manage seasonal fertility. There are certain animals that only give birth in particular seasons. With improvement in animal breeding and genetics and the use of technology, we can now manage such animals. We can also use moet and other methods. Moet is a method that is used to produce multiple ovulation in farm animals, and then the embryos can be recovered and then transferred into surrogate dams. Other areas where we are finding application which has helped in improving reproductive efficiency are in vitro reproduction. Also, embryo splitting and nuclear transfer, which has made it possible to move genetic materials from one cell to another. We also can now carry out embryo and semen sexing. So embryo and semen cryopreservation is also an area here we can now store embryos and semen under very low temperatures using liquid nitrogen for future use. Cloning and production of identical animals is another area of improvement. Another thing we can do is to carry out genetic performance evaluation and utilization of specific traits. And this is achieved through the planning of different breeding programs and analysis of performance records, which are now stored computerized, and their outstanding cells are listed so that we can easily access such information, and we can use semen of such outstanding cells for artificial insemination. Progeny testing is also another thing that we can perform so that we can use the result to formulate breeding plants. Another area is the application of marker-assisted selection, MAS. Marker-assisted selection is the use of certain segments or portions of the chromosome, of the DNA, that have been linked to certain phenotypes or performance in individuals. So on that basis, individuals can be selected on the basis of how they perform. So it is also used for parental verification and use it to know who or the parents of specific animals. We can also use it for sexing and in molecular breeding. We can also use it for transgenesis, which is basically the transfer of useful genes across species. Can move it from plants to animals and vice versa, or from one animal species to another. So marker-assisted selection is helping us to produce genetically modified organisms, although there are controversies pertaining to GMOs, as the case may be. So individuals that have higher yielding ability and greater resistance to diseases and pests, for example, are selected. So artificial insemination is one methodology that has greatly impacted on the improvement of animals. This is applied mostly through embryo transfer, especially from animals or proven genetic sources, and also in the area of sexing of embryos, gametes, cloning, and of animals, and through gene technologies that can be applied to a large number of embryos. These embryos are usually obtained by mating outstanding sires to several females. Another area we are making impact is in animal nutrition, and this is through the use of biotechnology to produce plant materials that will provide qualitative feed to animals. For example, we are now able to produce forage plants that are able to withstand stress, diseases, and pest infestation. Also, be able to improve plant quality, post-harvest storage, so that we can have good quality feed resources to feed our animals, particularly the ruminants. We can now even manipulate rumen microbes to an extent as to enhance better digestibility of hitherto unavailable plant products. So there is also the application of feed components and additives that have been synthesized biotechnologically, and these are incorporated into feed in order to enhance the performance of farm animals. There are certain enzymes such as phytates which have been produced, which are now helping us to break down plant material, releasing very important nutrients such as phosphorus in plant parts. These are now becoming more available to animals so that they can use them. At the same time, by breaking down the phosphorus, they are digested and they are not excreted in the feces. Another area where we have made progress is also in animal health. And here we are able to increase the productivity of and then the survival of farm animals. And this has greatly been improved through the application of the knowledge of genetics. There's improvement in disease diagnosis through the use of technologies such as PCR, which is acronym for polymerase chain reaction. This has helped us to localize genes on the chromosome. And it has led to better understanding of diagnosis and the effect of disease-causing genes and also germs. And through this technology too, we're able to produce vaccines and therapeutics through the application of knowledge of genetics for more effective treatment of largely preventable diseases. This collectively have led to low health risk in moving genetic materials from borders or across borders. Through gene transfer technology or biotechnological breeding, we've been able to now breed animals that are resistant to many pests and many diseases. So these are areas where improvement is coming into play, and through the application of genetic principles. There are other areas which might not have been captured in this material. Please read them up, like the use of surrogate cells